Hello viewers and learners of the Postgraduate Diploma in Environmental and Occupational Health. I introduce myself as Dr. Sushmita Bhaskar, working in the discipline of Environmental Studies at the School of Interdisciplinary and Transdisciplinary Studies, Indira Gandhi National Open University. Today we will discuss about the basics of ecotoxicology, which forms a part of the course MEV 004 Environmental Toxicology. So, we have seen the introduction to the toxicology and some of the sources of the toxicants and the mode of action and the distribution of these toxins. Also, we have discussed where these toxins can even be stored for years together in our body. So, that way there can be certain problems to the organ toxicity, systemic toxicity, and finally unless these toxicants are eliminated or taken out of the body, only then we can lead healthy lives. So, now let us see the basics of ecotoxicology. Ecotoxicology, this term was first defined by René Trohat in 1969. As we see, it combines two subjects, ecology as well as toxicology. So, that is ecological sciences and also the toxic effects, the toxicants and all the subject and the discipline that is dealing with the toxic effects of the different substances on the environment as well as on the human beings. Now, ecotoxicology, this aims to assess the impact of chemicals and several other toxicants not only on the individuals but on the populations and on the entire ecosystems. So, it is on at the population level, at the community level, at the human being, the entire study and the testing that comes under the domain of ecotoxicology. Initially, the important tools of environmental toxicology, these were the detection of the toxic residues in the environment or in the individual organisms. So, that is in the environment we were seeing for certain pesticide residues or certain other toxicants. It could be even hazardous waste materials, and then also in the individual organisms, like for example we take the rodents, we take the white mice, or we take the fishes. These are all used for testing of the toxicity of the chemicals. So, basically there can be plant assays, there can be animal assays, and then also on the human beings we can collect the blood and we can also see certain other problems and the toxic effects that are exerted at the different levels. The subjects of ecotoxicology, these were developed as a need to measure and predict the impact of the pollutants on populations, on communities and the whole ecosystems rather than on the individuals. So, when we are discussing about a terrestrial ecosystem or an aquatic ecosystem, so there we are studying on the entire population, the community and the individuals who are going to be affected by that toxicity which is caused in that entire ecosystem. Now, the word ecotoxicology can also be defined as the study of the harmful effects of chemicals upon the ecosystems. This was given by Walker et al. In 1996. It can also be defined as the field of study which integrates the ecological and the toxicological effects of chemical pollutants again on the population, the communities and the ecosystem with the fate of such pollutants in the environment. So, when we talk about fate, that means it is a transport, transformation and breakdown of these pollutants in the environment. This definition was given by Forbes and Forbes in 1994. At an individual level, the toxicants may disrupt the biochemical, the molecular and the physiological structure and function of the living organisms, and this can have serious consequences. Now, toxic substances, they are usually investigated using toxicity testing techniques. So, that is they aim to test the dose or the concentration of a particular chemical that is toxic, and this is done under controlled conditions in the laboratory, that is in vitro conditions. Now, the toxicity tests are mainly carried out using either plant assays or the animal systems. So, in the plants we can use the onion, that is the Allium cepa, then you can use Potulica oleracea. These are all plants that are used in order to test the concentration, the lethality of a chemical, and we can also see the genotoxicity and the cytotoxicity that is caused by certain groups of chemicals or the toxicants. And in this for example, if you take the Allium cepa and then when you squash the cells and then you add the stain, Then after that you will see that the chromosomal aberrations can be seen. You will see in the different mitotic stages, whether it is a telophase stage, the anaphase stage, you will see that either the chromosomes will be sticky or they will be bundled up together, or certain other chromosomal aberrations. There can be deletions, there can be insertions, you can find ring chromosomes. So many of the different types of anomalies you can see, and that is all due to the toxicity of that chemical that is exerted on the plant or the animal system. Now in the same way, you can also do it on the animal system. So you have the white mice. In this you can test the bone marrow of the mice, and then using certain stains like the Giemsa stain and so on, then when you examine them under the microscope, then you can see the different types of the changes, the aberrations that have taken place due to the toxicant or due to the toxicity of that on the animal. Then you also have to compare that with the normal system. Like if you are taking the toxicant at different levels, you can also give that at at different dose or the concentrations which you are applying, and then you take a normal system, you have to standardize it, and then you will come out with the data. So not only in the plant or in the animals, the higher animals like the white mice, but you can also do it with the nematodes. You can also do the test with the earthworms. You can also do it with the pilar species, that is the mollusk, and so on. So you can take any of these animal, the test systems, in order to test the toxicity of a certain chemical and what kind of effect they will have. In the same way, you can also test it on the fishes, on the algae. So in the fish especially, you can go for the tissues of the fish and where that will be accumulated. So in the tissues, from that you can see how much of concentration of that will be accumulated in the fish. So these are all certain tests that are being done in the ecotoxicological laboratories. Therefore, ecotoxicology, as defined by Butler in 1978, it is concerned with the toxic effects of chemicals and physical agents on living organisms, especially on populations and communities within defined ecosystems. So it includes the transfer pathways of those agents and their interactions with the environment. It is also the science that seeks to predict the impacts of chemicals on ecosystems. So these are all the different definitions that have been given by different scientists. And also Walker et al. In 1996, they said that it is a study of harmful effects of chemicals upon the ecosystems. Xenobiotic. Xenobiotic itself means something which is foreign to a body or something which is strange, which is unusual. So also referred to as a foreigner or a foreign particle. So this is used to describe the compounds that are foreign or not normal to a particular organism, and a chemical which is normal to one organism may be allergic or it could be a foreign substance to another. For example, biological toxicants like mycotoxins, aflatoxins, some may be allergic to peanut butter. Okay, so because it is producing some kind of a allergic reaction, so it may be normal to one person but it could be allergic to another. In the same way, you have the gluten intolerance. So it can be normal. So certain wheat products and wheat containing or the food products which are containing gluten. So that is why those people need to be aware and they need to see the food packets and the labels whether that contains gluten or not. Otherwise it can cause hives and several other problems in the system. So xenobiotic may be naturally occurring, it can also be man-made compounds. Some of the important issues which have influenced the development of ecotoxicology. The first is DDT, dichloro-diphenyl trichloroethane. So this has affected people globally, and the DDT, it was first when Rachel Carson, which she came out with the publication of Silent Spring in 1962, and it is a landmark in the public awareness for the potential damage to the human and also to the environmental health from the man-made toxic substances. So this was actually, she challenged the practices of the agricultural workers where the DDT was sprayed, and she saw that those robin birds and the birds were actually disappearing. Later on we also found that there was thinning of the birds' egg shells, and all this was due to this pesticide which was applied in a large enormous manner. And again, this is a persistent organochlorine compound and it remains in the environment for several periods of time, and it also accumulates in the environment. Cadmium, which is a heavy metal in Japan. So this again we have the nickel cadmium battery sectors and several other industrial sectors, and in the agricultural fields it was noticed, and the rice containing cadmium was noticed, and people complained of several joint problems, known as the itai. Mercury in Japan, this was also released from the industrial sectors, where people complained of unexplained ailments like neurological symptoms. They had visual disturbances, then they had paralysis of their hands and their limbs and so on. So this was also toxic, because the inorganic mercury was converted into organic methyl mercury in the aquatic ecosystem by the methylation process by certain anaerobic bacteria. So bacteria made this process, and then all forms of mercury, inorganic, organic or elemental form, these are very toxic to the humans, to the environment. And so the fishes started accumulating this mercury concentration, and since this area was the people are highly dependent on the seafood and on the oysters, on the shellfish and on the fishes, the crustaceans. So the people who consumed these fishes that were contaminated with the mercury, they developed this strange disorder, and since it was noticed in the Minamata Bay, the disease was also referred to as the Minamata disease due to acute mercury and chronic mercury poisoning. PCBs in Japan and Taiwan, this was also an issue, important issue, which has influenced the development of ecotoxicology. Dioxins, which are also the POPs, persistent organic pollutants, they also remain in the environment for several years, and they are released by several processes, by combustion, industrial processes and so on. So dioxin also have several negative effects on the environment, and since they are persistent, they remain for long periods. The contamination of the pristine environments like the Arctic environment by the atmospheric transport of the organohalogens. So all these are certain important issues which we need to know. Now dioxins in the environment, that is specially I am giving a focus. The sources come from the combustion sources like diesel vehicles, coal fired utilities, wood burning and the cement industries. Again from the reservoirs like forest fires, landfill burning, then biochemical and photolytic processes. It can also come from industrial sources like the paper and the pulp mills, from the power boilers, from the chemical industry, from the metal industries. It can also be generated from the incineration sources like the sewage sludge, municipal waste, medical waste and also hazardous waste. So these are all the several sources from which dioxins come into the environment, and they can persist and cause several issues with the human health. The fate of the chemicals in the environment and within. So now that we have discussed some chemicals which are causing irreversible effects in the humans and also in the other living organisms and on the environment, ecotoxicologists are concerned with the movement and the fate of these toxic chemicals in the living organism and in the entire ecosystem. Therefore, the source, transport, modification and final fate of the pollutant are very important in each study. At the organism level, the mechanisms involving the uptake of the toxicant, excretion of the toxicant, sites of action where this toxicant is going to create its toxic effect, the metabolism and the storage, these are also very essential points which need to be pondered upon. Toxicity testing and the regulation and the release of the toxic chemicals. So we all have gone to the laboratories, environmental and ecotoxicological or any of the pharma laboratories. So the toxicity test involving pollutants are based on the test on the individual organism in artificial test situations in the laboratory. So these are carried out on a large scale. These tests are cheap, reliable and easy to perform, and the toxicity testing can be done using single species. They provide useful information for the regulation and the registration of the toxic chemicals, because otherwise how will we know that this thing is toxic for us, or this chemical is toxic for the animals or for the plant or the human system. Testing methodologies. So there are different ecotoxicological and biodegradation tests for the chemical, agrochemical and the pharmaceutical industries. So these include the bacterial toxicity test, we have the algal growth test, acute toxicity test with the Lemna minor, acute and reproduction test in Daphnia magma, acute toxicity test with the marine copepod Acarcia tons, oyster embryo larval toxicity test, acute toxicity test with the marine invertebrate, earthworm toxicity test with the sediment dwelling organisms such as Chironomus or the Lumbriculus. Then we also have the test with the freshwater and the marine fishes. So, these are all the several toxicity tests that can be performed on the several algal, bacterial, and the plant systems. Endpoints, dose and the concentration. So, these are also important terminologies which are important in the ecotoxicological studies. There are many different ways in which toxicity can be measured, but they are usually assessed relative to a particular outcome or endpoint. So, majority of these 9 toxicity tests which we discussed, they measure the number of organisms that are killed by a particular dose or a concentration of the chemical that is being tested in the laboratory. So, the dose, this is used where the dietary dose of a test chemical can be accurately determined. For aquatic organisms, the test measure the concentration of the chemical in the surrounding water or the medium. Now, some more tests which can be used and then to extrapolate the data from is LD50, that is median lethal dose, which kills 50% of the population. Then we have the median lethal concentration, which is given as LC50. ED50 and EC50 is the median effect dose and concentration, which is the dose that is produced a defined effect to the 50% of the population, that will produce a defined effect to the 50% of the population. Then we have the no observed effect dose or concentration, no observed effect level, because these are used as the levels that the chemical does not cause a statistically significant difference than from the control. Then we have the lowest observed effect dose or the concentration. See, these are the various terminologies that are used. Then hazard and the risk. So, these are two different definitions. Toxicity data is used to make assessments of the hazard and the risk that is posed by a particular chemical. So, hazard is a potential to cause harm and risk is the probability that the harm will be caused. Hazard is a product of exposure and toxicity. Therefore, a compound with moderate toxicity but very high exposure, they may cause more damage than a very toxic chemical with a very low exposure. So, please keep these points in mind. Risk. This is usually defined using a predicted environmental concentration and the predicted environmental no effect concentration. So, information on the movement of the behavior of the pollutants in the environment are used to calculate the predicted environmental effect or the concentration, whereas the data from the toxicity testing must be extrapolated for calculating the PNEC, that is predicted environmental no effect concentration. So, therefore, only then you can find out the risk of the specific toxicant to an individual. Biomarkers, these are defined as a biological response to a chemical or chemical that gives a measure of exposure and sometimes also of toxic effects. They can be divided into biomarkers of exposure and of toxic effects. Examples of biomarkers. So, they range from the inhibition of the acetylcholinesterase in the nervous system of animals to the thinning of eggshells in birds. They can help establish the gaps between the laboratory and the field by giving direct evidence of whether or not a particular animal, plant or ecosystem is being affected by pollution. Immunotoxicology. So, this is the study of immune dysfunction resulting from the exposure of an animal to a xenobiotic. The immune dysfunction, this will include immunosuppression, allergies, autoimmunity or a number of inflammatory based diseases or pathologies. In the embryo, for example, fetus, the neonates, this study is referred to as developmental immunotoxicology. For most of the toxicants, the embryo and the fetus, they will show a heightened sensitivity compared with that of an adult, because they are very sensitive and they are also being formed in the very primitive stage when compared to the adult. Immunotoxicology. So, this is relatively a new interdisciplinary scientific field, and this focuses on the identification and analysis of the chemicals in a broader sense, also to the physical and the biological factors of the environment. This can cause unwanted and incidental immune modulations. There is also a term known as immunopharmacology, which investigates the immune modulative effects of various substances that are applied intentionally for therapeutic purposes. Therefore, immunotoxicology investigates the properties of new immunotherapeutic pharmacological products. They are prepared via recombinant DNA technique and with regard to their immunotoxic potential and the safety of their use. This includes the interleukins, interferons, growth factors, anti-inflammation drugs, neuroendocrine hormones, neuropeptides and so on. So, it also deals with the immunotoxins, the substances isolated from bacteria and plants, and the general property of which is the inhibition of proteosynthesis, that is diphtheria toxins, the Pseudomonas exotoxin, then ricin, ribosome inactivating protein and others. So, this is an idea of the immunotoxin and immunotoxicology studies. Substances which are exhibiting the immunotoxic effects. So, immunotoxicants are the factors of the external environment and they can cause significant changes in the immune mechanisms both in the human and in the animal systems. Then there are immunomodulators, which are a group of substances of the different origin which also affect the immune system. In the environmental toxicants, they can include the pesticides, industrial emissions, drugs, then we have the radiations, the physical factors like UV light, especially the UVB light, electromagnetic fields. All this include the environmental toxicants which can exhibit the immunotoxic effects. So, the action of the immunotoxicant, this will result in reduced reactivity of the immune system. That means it is causing a certain kind of immunosuppression like the PCBs, the lead or the cadmium, and it can also cause excessive reactivity like in the case of polycyclic aromatic compounds, organophosphate insecticides and so on. Some substances like nickel and mercury, they can also lead to autoimmune diseases. The susceptibility of the immune system to immunotoxicants, this depends on the properties of the respective chemical and the complex nature of the immune system, that is the antigen recognition and the processing, then cellular interactions, cooperation, regulation and amplification, activation and differentiation of the cells, and also production of the mediators by the various cell types. Therefore, the interactions of xenobiotics with the immune system, they can also be affected by the additional factors such as malnutrition, stress and genetic predisposition. So, from the point of view, there are several mechanisms of immunotoxicants that we recognize, that is there is systemic and the local effects, and there are also some selective effects on the target cells, that is the beta lymphocyte, B lymphocytes, the T lymphocytes and also the macrophages. Systemic immunosuppression action. This is affecting the activity of the immune system as a whole, and that can develop due to the action of various chemical agents. For this action alone, there were certain studies, detailed studies that were carried out in the rodents. And what were the observations that the scientists made? They observed altered weight and histology of the lymphoid organs, quantitative changes in the cellularity of the lymphoid tissue, peripheral blood leukocytes and bone marrow, impairment of the cell function at the effector or regulatory levels, increased susceptibility to the infectious agents or transplantable tumors. Possible mechanisms for the xenobiotic. They can affect the immune cells firstly. They can affect the antigen distribution and or persistence, influence the production differentiation of precursors immunocytes. They can influence activation threshold of immunocytes to antigen growth and the differentiation factors. They can affect the production and release and the metabolism of certain substances like the lymphomonokines, the phytoprotein modulating the immune cell function. They can alter the functional capacity of the platelets and the neutrophils. They can also affect the central nervous system endocrinological homeostasis and balance. Hypersensitivity mediated by the xenobiotics. So, these are all certain technical terms that we have discussed, because we should know the mode of action and at what level even the immunotoxicants can have. Now, the hypersensitivity, this is what first we are observing when we are contracting or the exposure to the contact dermatitis to any kind of a metal or any environmental toxicant. So, contact hypersensitivity, this is induced by the xenobiotics. The primary exposure, it can develop for several days, weeks, it can also last for several years. For example, you can find eczema, erythema, there can be swelling, there can be some kind of the pus formation, pruritus, it all will predominate in the acute phase. Then the contact hypersensitivity, this depends on additional physiological as well as pharmacological factors. It depends on the permeability of the skin, and also age of a person, which affects directly the antigen dose uptake. Now these sensitivity and the sensitive reactions, sometimes people can be even allergic to certain earrings or the metal jewelry that they wear, bracelets for example. They can have several metal induced problems and hypersensitivity. So it could be caused by pains from the polycyclic aromatic compounds, from pesticides, from different chemicals like the formaldehyde, epoxide, drugs like the neomycin, and also certain plant products. Then contact dermatitis can be from the nickel jewelry, metal frames, dog collars, and so on. Nickel will easily dissolve in the weakly acidic environments of sweat, and its salt will have the high ability to bind to the proteins, including albumin and protein of the complement system, which can create the conditions for the development of these hypersensitivity reactions. Other hypersensitivity reaction, they include the pneumonitis that is induced by inhalation of various allergens like the farmer's lungs, the furrier's lungs, the pigeon breeder's disease, cheese washer's disease, and so on. So in this slide I have given you some examples of the hypersensitivity reactions, like you have the farmer's lung, which is actually caused from moldy hay. Then the sawmill workers lung disease formed from the moldy wood dust. Then the bird farmer's lung, proteins that are excreted in the bird droppings. Then you have the mushrooms washers lungs, and also the malt workers lung. Both these are due to the spores and the molds, the moldy malt which is inhaled, the spores which are inhaled by the farmers. Then you have humidifier lung, where there is contaminated humidified water which is persistent. Cheese washers lungs, this is caused by the Penicillium species. Then you have the hard metal washers lung. So here again you have the hard metal dust, cobalt, and so on. So there are so much of the different type of the moldy dust and the contaminated settings in farm and agricultural environment. So even these products, agricultural produce, also can cause certain pneumonitis and toxicological disorders of the lung. Autoimmune diseases. So one of the characteristic features of the immune system is the recognition and development of immune response to various xenogenous antigens, and these are present in the external environment and the absence of immune response to the own tissues. So some degree of autoreactivity is a normal feature of the immune system, because some proteins, many carbohydrates, lipids, nucleic acids, these are all potential antigens that are capable of inducing humoral and cellular immune response. An impairment of the immunotolerance caused by various factors. Some more diseases of the autoimmune diseases which are caused due to environmental toxicants are autoimmune glomerulonephritis. This is specially caused by mercury and gold. Then we have the IgA deficiency, which is caused by diphenyl hydantoin. The production of autoantibodies that was induced by the Spanish toxic oil incident. Scleroderma resembling lesions that has been induced by the vinyl chloride, which is used in the plastic manufacturing sectors, where they use vinyl chloride even for the flooring and in the rubber industries. Then autoimmune hemolytic anemia. This is an interaction of the antibodies with erythrocyte bound penicillin or the pesticide edieldrin. So these are the several autoimmune problems and the toxicity, especially with reference to immunotoxicological studies, where the environmental toxicants, heavy metals, vinyl chloride, pesticides, we have discussed dioxins, then the dieldrin, DDT, the same way certain other the chemicals like the vinyl chloride, also certain plant and the biological products. These all can cause ecotoxicology at the plant, animal, in the entire ecosystem, and finally the human beings are affected irreversibly. And many of the diseases, even these autoimmune diseases, they can be life threatening. It can cause several diseases which have no cure at all. So that is why environmental toxicants, these need to be thoroughly investigated, they need to be understood, and we need to go in for organic food products, and even avoid the use of certain heavy metals and heavy metal jewelry and certain other the paint which is actually giving out the fumes of all these chemicals, which can induce certain other autoimmune diseases. So I hope you have had a good understanding of this subject, that is the basics of ecotoxicology and with special reference to the immunotoxicology. Thank you for your patient listening.